Hello, welcome to another screencast. We'll be discussing Mendel and his experiments. And by the end of this, you'll be able to describe and explain why Pisum sativum, which is the pea plant that he used, made a good organism for the genetic studies, hereditary studies. Remind yourself about Mendel's experiments, his expectations or his hypotheses, if you will, and then what his observations were or what his actual results looked like in terms of monohybrid crosses, a test cross, and a dihybrid cross. So again, this all should be a review from Bio 1. And in number C down here, be able to explain how Mendel used mathematical routines. And these mathematical routines and logic is what we're talking about with the rule of multiplication, the rule of addition, and that exercise, that tutorial I had you do about statistical analysis, etc. Right? So using how he used math to explain his observed inheritance patterns, and then focus on the chromosomal evidence. This is the modern evidence that supports and validates Mendel's experiments that were conducted back in the 1860s before even cellular work was even really truly understood. Okay. So here's Mendel on the left. On the right is his abbey. He was a monk. These are his little pea plants growing in one of his gardens. And Pisum sativum made a good organism of choice because he could control the mating. So here you see him in a cartoon version of him anyway, cutting off the stamen here, or the male parts, the anthers, removing those so that there's no chance of any self-fertilization or that pollen getting anywhere. And then here you see him moving the pollen grains, which contain the sperm, from one plant to the next. And so this is the fertilization, the pollination right there, which then leads to fertilization. So he can control the matings. He is performing some sort of artificial selection here, right? He's selecting which organisms he gets to cross. Another reason that this is a good organism is that when they reproduced, they yielded lots of offspring. So the peas, these seeds, are the next generation. Right? So the seeds are offspring. Right? And you produce many offspring from one mating, right? And so your sample sizes are relatively high. So this eliminates any sort of random error issues there, and can perform some sort of mathematical routines, some statistical analysis, then to support the findings. And the other thing that makes this a good organism of choice is that there are many easily observable phenotypes, as you see on the table on the right here. So you can look at the flower color, self-explanatory. The flower position is either axial or terminal. The flower color was purple or white. And so you have these, right? These are OR, so these are known as discrete characteristics or discrete phenotypes here. It's either or. You didn't have like a light purple. This is like white, but like a little purple. You had either purple or white. It was obvious. The flower position was either axial, terminal, right? Seed color, seed shape, pod color, stem length. So Mendel knew his pea plants here pretty well. Easy to care for. Plant them in the ground, a little bit of sun, a little bit of rain, and they're going to grow pretty easily. You can plant a lot of them in a small space, so you can do these experiments over and over. The turnaround time on these is relatively short, so you can get lots of generations and able to follow and track a trait through the several generations. So some of the experiments that he did, he knew that some of these pea plants were true breeding, right? He would take true breeding parents, and that means that if you had a plant that produced purple flowers and you crossed with purple flowers, you're always going to get purple flower offspring in the F1 generation. So it had your P generation for parent generation, so this is where he was starting with true breeding. His F1 generation, and the F stands for filial, right? So this filial generation, and you would then cross those filial generations. So you take organisms from the same generation and you mate them together. So you cross these, and that's how you get the F2 generation. Right? So when you cross the F1s, you got F2s, and they were all purple. He did the same thing for the whites. Right? Here, so white crossed with white in the P generation got you all white in the F1. When you did the F1 cross to get the F2 generation, those are all white. So he knew that he had true breeding. He knew he had true breeding purple, true breeding white flowers. So the next logical step was, okay, well, if I cross true breeding with white, as seen over here, right? If I cross true breeding purple with true breeding white, the expectation for the hybrids, the F1 generation down here, should be that I get, whites and purples. So again, this was his expectation. But as the title of the slide sort of indicates here to you, Mendel's subsequent breeding experiments of the observations did not match his expectations, right? So his expectation was to get white and purple, right? But he did not see this. This actually were his observations over here when he crossed true breeding purple with true breeding white, is he got all purple plants. And this made him say, what the heck is going on here? I don't understand, and he kept doing this, and so he was reasonably sure, okay, when I cross true breeding purple with white, I'm always going to get purples. And so this led him to coin the term that the purple phenotype was dominant to the white phenotype. And so then he said, okay, well, let me cross these F1s, right? So now that I have these two purples, right, if I cross two purples here, then I should be getting all purples, right? And that is not actually what he saw in the F2. What he saw in the F2 was the classic, of course, 3-to-1 ratio. He got whites and some purples, right? And so this made him at first go, what the heck is going on? I got purples and whites when I was expecting to see all purples, right? But then what he notices, a keen eye in Mendel, is that he counts, right? He sits there and counts all these offspring, and there are 705 plants that had purple and 224 that had white flowers, and he saw that this, okay, wait, this is a ratio. This is in a pretty close ratio, right? Incidentally, this over here, if he had gotten this, which he did not, if he crossed these two, he would expect to see, equal numbers of those. But again, this never happened. He didn't do that cross there, right? So he sees this 3-to-1 ratio. This 3-to-1 ratio leads him to say, oh, okay, this is that the purple is dominant to the white, right? And he then goes on to do this experiment with several other of the discrete phenotypes, right? And so here you see his F2 generation numbers, right? So here's his 705 to 224. And that ratio wasn't quite exactly 3-to-1, but it was 3.15 to 1, right? When he did flower position, he isolated those matings, right? 651 to 207. So Mendel's busy counting, right? This is 3.14 to 1, right? Look at this, 6,000, right, and 2,000 roughly, right? 5,000, 1,000, 380, so he's spending a lot of time counting offspring here in the F2 generations. And what he also noticed is that, wait a second, the larger the number, so this is the largest sample size I have here, look how close he was to that exact 3-to-1 ratio, right? These other ratios, not quite as close. In fact, the one that's off the farthest here, this 2.82 to 1, right? This indeed was the fewest number that he counted, so his sample size was pretty low there. So this should tell you something about sample size, right, and expectations and observations here. And this will come out in the Fly Lab when we talk about this a little bit later, right? So what he does then, he goes and he throws some notations on here, and this is where we get the big letter, the capital letter, and the lowercase letter. So what he was saying is that these true breeding parents here, big P, these parents donated a big P, right, to the mating. These parents over here donated this little p to the mating, and then you get these hybrids down here, right? So this is a hybrid. All of these F1 generations were a hybrid. Right? And this is where you get this monohybrid cross. You're looking at one trait, and when you do the monohybrid cross, that would take another purple flower from this generation that was also heterozygous there. And so you would cross these two, and this is the F1 cross to get the F2 generation. This is the monohybrid cross right here. Monohybrid. You're looking at one characteristic, and you're crossing the hybrids here. These are hybrids because they are the result of two individual parents there. So just monohybrid cross. The probability then of this parent producing a gamete that has the big P is 1 out of 2, or 0.5, and the probability of producing a little p from this parent is 1 out of 2, or 0.5. Right? And so if there's a way then to predict here. So these are the F1 sperm possibilities, right? So this is 0.5 probability, 0.5 probability from the F1 ova, right? Probability that hybrid parent produces a little p is 0.5 and 0.5. The probability that this independent event here and this independent event there, that is the production of this gamete and the production of this gamete, are independent events, right? which gamete this parent produces is independent of which gamete this one produces. So the probability of these two independent events happening at the exact same time, that is fertilizing, here is the product of each of their individual probabilities of happening at all. So use the rule of multiplication here. So 0.5 times 0.5 is 0.25. Right? Probability of getting this is 0.25. Probability of getting this is 0.25. Probability of getting this is 0.25. So the probability of getting a heterozygote is actually both these, because you can get the heterozygote in more than one way. So the probability of getting a heterozygote there is 0.5. But the probability of getting a purple offspring, a purple one here, is 0.25, 0.25, 0.25. These are all purple, so you get a 0.75. Right? So this is 3 to 1. So you would expect 75% of the offspring to be purple, and you'd expect 25% of the offspring to be white. So if you had 1,000, offspring, you'd be able to you would say that you would expect 750 to be purple, and then 250 to be white. Well, what if you had like 6,250? Right? Well, you'd take this times 0.75, and that would give you how many were purple. Take that times 0.25, and that will get you how many were white. Right? So the Punnett square here and the math, the logic is working out. So he says, oh, okay, this must be it. He comes to this conclusion that these parents are giving something to the offspring, right? And this was his notation. This is his way of sort of tracking that, right? I would recommend sort of clicking on this this here and working those problem sets, those tutorials, to see if you understand that. The test cross. The test cross is a way to determine the genotype of the dominant phenotype. Remember that the dominant phenotype was purple, right? But the genotypes for purple would be big P, but also big P, little p, right? So how do you know if the purple parent was this or this? How do you know if the purple parent was homozygous or if the purple parent was heterozygous? Well, what you do is you do a test cross. And a test cross is you take the dominant phenotype of unknown genotype. This is a definition that you need to know. Right? So dominant phenotype of an unknown genotype. And you mate that, you cross that with a recessive phenotype. And you know that genotype has to be little p, little p. And so if that purple parent was homozygous, then you would expect a 1 to none ratio. Right? You get all purples there. If the purple parent of unknown genotype was heterozygous, then you'd get a 1 to 1 ratio there. You get equal numbers of purple and equal numbers of white. Right? So when you want to know what the genotype of this parent is, I don't know what that parent's genotype is, you cross it with a homozygous recessive individual and then look at the offspring. You have to look at the offspring, you have to look at the ratio of the offspring to then tell you what this was. Right? So if it was that, then you'd see a 1 to none ratio. If the purple parent was heterozygous, then you would see that 1 to 1 ratio. Okay? Now, Mendel got a little bit more complicated than that. He was able then to start looking at 2 traits at one time, right, or 2 characteristics here. So now he looked at seed color and then the seed shape. Right? So he took true breeding yellow rounds and true breeding green wrinkleds, right? And he crossed those, right? And he then got all yellow rounds or yellow smooths here. And so this told him that yellow is dominant to green and that smooth or round is dominant to wrinkled. Right? So now his expectation was, okay, well, if I do this dihybrid cross, right, and cross these individuals here, then I would expect to see a 3 to 1 ratio, because this is what he was, seeing before when he did the monohybrid crosses. So now it should work the same way if he's doing a dihybrid cross, right? So his expectation was a 3 to 1 ratio. But again, as this big red X indicates, he did not see this, right? His experimental results contradict his hypothesis. His hypothesis, of course, being the expected results. The experimental results, of course, being the observed results. Right? So he has to go back to the drawing board and he says, well, let's stop a second. What did he actually see? He actually saw this. He saw a 9 to 3 to 3 to 1 ratio, right? So a 9 to 3 to 3 to 1 ratio. Right? And he saw this over and over and over again, and it made him say, oh, okay, there's a pattern here, right? I've got to explain this pattern. So he goes back to his drawing board and he says, all right, independent assortment. Right? What if the yellowness and the greenness were inherited separately, independently of the roundness and the wrinkledness? Right? And so when these parents produce gametes, right? When these parents here produced their gametes, right? That how the big Y and the big and how the Y's and the R's associated themselves or separated into the gametes was independent of one another, right? Well, that means I could, in the ova here, I could produce a gamete that has a big Y with a big R, a big Y with a little r, a little y with a big R, or a little y with a little r. Right? And each of those is equally likely, right? So this is a 0.25%, 0.25, 0.25, 0.25 probability. And then the sperm, the same thing, right? Each of these gametes here, 0.25, or a 1 out of 4 chance of producing them. Each of those is an independent event. And so what's the likelihood of getting this sperm fertilizing this ovum right here? Well, it's 1/4 times 1/4 there, right? 0.25 times 0.25, right? To get that. Right? So you would multiply this out, so that is 1/16, right? 1/16, 1/4 times 1/4, 1/16, and it turns out that this Punnett square has 16 boxes. So what's the probability of getting big Y, big Y, big R, big R? 1 out of 16. What's the probability of getting a yellow seed? Right? What's the probability of getting a yellow seed? Right? You have all these boxes, so this is yellow, but now you're talking about 2 at the same time, so yellow round. Right? The probability of getting yellow and round is 9 out of 16. Well, how do you get that mathematically? Right? What's the probability of getting yellow from these two parents? Well, the probability of getting yellow is that you have to be this, right? So this satisfies yellow, and this satisfies round, right? So what's the probability from this this Y, big Y, little y, big Y, little y cross of getting that? Well, that is 3 out of 4. Right? What's the probability of getting big R, anything there? That's also 3 out of 4. Probability of both of those two things there, right, is of course 9/16. You multiply this together, that's a 16. Right? 9/16, right there you go. Okay. So mathematical probabilities here supporting Mendelian genetics here. Right? So the genome turns out later on, fast forward, 100 years or so, and turns out that the Pisum sativum genome is contained on 7 chromosomes, right? And so the locus for these different characteristics are on the different chromosomes, right? So this might be here seed color. This is the seed color locus right there. And this is the shape seed, so seed shape locus, say, on this chromosome right here. Right? And since all these are on different chromosomes, they assort independently of one another. Right? So going back now, you can have this diagram, and we put now Mendel's alleles, right, onto the chromosomes. Right? And say that, yes, in fact, we are able to now take Mendel's notation and actually match that to the chromosomes, right? Where the seed color gene was on this chromosome, and the seed shape gene is on this chromosome. Right? And since those chromosomes are 2 different chromosomes, those chromosomes will assort independently of one another at meiosis 1. Right, which we reviewed in class. And so the likelihood of that happening here, of them assorting independently, you could get different arrangements. Now, because this was homozygous at both those loci, you don't get any difference there. But here in the F1 generation, the F1 cross, so when you do the F1 cross of this dihybrid, right, you see right here at meiosis 1, these are the 2 possible arrangements. And those 2 possible arrangements then if the arrangement 1 is here, you end up getting these gametes at the bottom over there. But this arrangement is as likely to happen and will happen about half the time. And so your other half of your gametes, right, are over here when you get this arrangement. Right? So Mendel, way ahead of his time, knew nothing about chromosomes, right? Yet here's the chromosomal evidence to support and to back up Mendel's mathematical conclusions, his logical deductions, etc. Right? So this is just kind of an interesting thing here to me. Mendel used the term character and trait, right? Turns out that his character, so the character would be like, seed color, right? And then the trait for seed color was either yellow or green, right? And it turns out that what we now call the characters are actually the genes, right? And so the gene is at the locus. So at this locus right here would be the locus for seed color gene. And then the trait would be the allele, right? So if you zoomed in here or here, you'd see a DNA sequence. And one DNA sequence gets you a yellow protein that's made, and the other DNA sequence gets you a green protein that's made. So the DNA sequences here were different. And it struck me like, how am I going to remember this? Like Mendel knew nothing about chromosomes, knew nothing about DNA or genes or anything like that. And it dawned on me when I just wrote this out, and I saw like, oh, Mendel's characteristics turned out to be genes, right? And his traits turned out to be alleles. So the characteristics are today's genes, and the traits were the actual, what we call alleles now. And it just struck me that those are the 4 nucleotide bases in DNA, and they are, complementary to one another, right? C and G and T and A there. So just, cool, I thought. Anyway, so that's a little bit of a long one there, but a quick review of basic Mendelian genetics right here. So a little bit about why Pisum sativum was a good organism. A review of Mendel's monohybrid crosses, his test crosses, and his dihybrid crosses. A little bit of new stuff here, adding the mathematical routine. So this is the rule of multiplication, the rule of addition, and those probabilities. And then the chromosomal evidence to support Mendel's conclusions and his experiments, right? Now, the rest of this is what you need to be able to do. So here's some practice. There's a whole bunch of questions here from a problem set, interactive questions, right? Another question. So I would recommend sort of going through these to test and see if you really understand how to do genetics problems, understand that whole rule of multiplication, the rule of addition, what the monohybrid crosses, the dihybrid crosses, right? Probabilities, etc. Right? So there's plenty of things to sort of work on. You don't have to do every single one, but maybe dip in there and test your knowledge there. Okay.